This video explains how to perform transmission electron microscopy, also known as TEM, on the Drosophila embryonic heart. The heart has emerged as a valuable model system for studying cell migration, cell-cell adhesion, and cell shape changes. This technique allows visualization of heart lumen shape, as well as ultrastructural components such as cell membranes, cell-cell junctions, and basement membrane. To begin this procedure, a fixation solution appropriate for transmission electron microscopy of Drosophila embryos is prepared. Next, a 0 to 20 hour collection of decoriated embryos are placed into the fixation solution. When finished, embryos are removed from the fixation solution and hand-devitrinized while attached to double-sided tape in a petri dish containing buffer. Devitrinized embryos float in the buffer and are recovered with a pipette. Then, embryos are post-fixed and undergo a dehydration gradient. Once infiltrated with resin, they are transferred to an embedding mold where they are aligned. The resulting block is cut with a diamond knife. Sections are picked up on copper grids, stained, and examined.
Hi, I'm Nadine Saplot from the laboratory of Sunita Kramer in the Department of Pathology and Laboratory Medicine at the University of Medicine and Dentistry of New Jersey at Robert Wood Johnson Medical School.
I'm Raj Patel. I'm the electron microscopist, also from the Department of Pathology. Today, we will show you a procedure for preparing and imaging Drosophila embryos for transmission electron microscopy.
We use this procedure in our laboratory to study Drosophila heart morphogenesis. So, let's get started!
Before handling the Drosophila embryos, freshly prepare fixative solution for the heptane permeabilization method of embryo fixation. In a fume hood, pipette 2 milliliters of a freshly made solution containing 12.5% glutaraldehyde in cacodylate buffer into a glass scintillation vial. Add 8 milliliters of eta-heptane and shake vigorously. Then wait 1 minute, allowing the two phases to separate. The upper phase contains the eta-heptane saturated with glutaraldehyde, which is the fixative solution. Pipette the upper phase into a clean vial and set it aside. To collect 0 to 20 hour embryos, use an embryo collection chamber, which is a small basket with a removable mesh insert. Begin by removing the grape juice plate containing the embryos and remove any dead flies. Then, using water and a paintbrush, loosen embryos from the grape juice plate. Transfer embryos into the embryo collection chamber using a squirt bottle containing water. Rinse the embryos of any excess yeast from the grape juice plate. Remove the outer chorion membrane by soaking embryos in a 50% bleach solution for 2 to 3 minutes or until embryos float to the surface. Rinse thoroughly with distilled water and blot the embryos on a paper towel. Using forceps, pick up the mesh insert, which should now be covered with decoriated embryos. Place the insert into the fixative, allowing the embryos to fall from the mesh. Allow embryos to fix for 1.5 hours at 4 degrees Celsius while mixing on an orbital rocker. Once the incubation is finished, place the embryos into a petri dish lined with double-sided tape, taking as little of the fixative as possible. Shake the dish to spread the embryos into a single layer, and then place it in the chemical hood to allow the heptane to evaporate. Once the embryos have dried, cover the embryos with buffer containing 0.1% tween. Hand-devitrinize late-stage 16 embryos under a dissecting microscope with a blunt tungsten needle. Embryos that have been devitrinized float and can be recovered with a Pasteur pipette. Transfer embryos to a 1.5 milliliter microfuge tube. Rinse embryos in 0.1 molar cacodylate buffer and then post-fix in 1% osmium tetroxide for 1 hour at room temperature. Following the incubation, rinse the embryos with cacodylate buffer once again. Dehydrate embryos in a graded series of ethanol and acetone. Start with 50% ethanol for 10 minutes, followed by 70% ethanol for 10 minutes. Then dehydrate in 90% acetone for 10 minutes, and finally in two 100% acetone steps of 10 minutes each. Remove the acetone. Begin the infiltration process by adding a 1-to-1 mixture of Epinspur resin and acetone. Ensure that the sample is under vacuum pressure and microwave for 3 minutes. Maintain vacuum pressure for an additional 5 minutes. Exchange the resin-acetone mixture with 100% resin and microwave again under vacuum pressure. Exchange the resin and microwave under pressure a final time for a total of 3 infiltration steps. Transfer the embryos from the microfuge tube to a silicone embedding mold. Align the embryos into a row such that the posterior end of the embryo aligns with the tapered edge of the mold. Bake in a 70 degrees Celsius oven overnight, then remove the resulting block for sectioning. Use a microtome to cut 1 micrometer sections into the embryos. Once the block has been sectioned through and the embryo has been reached, use a loop to move a single section onto a glass slide to determine whether the proper depth for visualizing the heart has been achieved. Prior to visualization, briefly heat the section on a hot plate and stain the section with methylene blue by first adding a drop to the slide and then briefly heating again. Rinse with deionized water and examine the section under a microscope. At the desired depth of sectioning, the heart lumen should be recognizable with a light microscope. To satisfy space constraints, retrim the block to include a maximum of 5 embryos in cross-section that show the embryonic heart. Cut 90 nanometer sections using the microtome. Pick up multiple sections on a copper grid. Stain the grid with 3% uranyl acetate for 10 minutes, then rinse 3 times with distilled water. Now stain the grid with lead citrate for 2.5 minutes in a chamber containing sodium hydroxide pellets to create a carbon dioxide-free environment. After the incubation, rinse 3 times with distilled water. Examine the sections with an electron microscope at 80 kilovolts and photograph with a CCD camera. In order to determine whether the sectioning depth and staging were properly performed, it should be apparent that a mature heart lumen has formed between the 2 cardioblasts of the dorsal midline. To determine whether cell membranes are intact, examine the heart lumen for the presence of extracellular matrix and look for the presence of cristae and mitochondria.
So we've just shown you how to determine the ultrastructure of the Drosophila embryonic heart.
When doing this procedure, it's important to remember to properly stage the Drosophila embryos and to accurately measure the components of the resin and mix them well.
So thanks for watching and good luck with your experiment.